Hello everyone, we are shortly to start. We will be starting pretty much on time today, so we hopefully won't be keeping you too long. We've got a very interesting, good webinar coming up. So I hope that you'll be getting a lot from this, whether you're a new person to the ion channel field or a seasoned pro. I think Professor Bruce Bean's talk should have something for everyone. Professor Bruce Bean doesn't need to be introduced. Anyone who's been in ion channels for almost any length of time should know of Bruce's work. It's been seminal work since the 80s. He's been largely working in voltage-gated sodium channels, but also some great work where I first saw Bruce's work in voltage-gated calcium channels and their characterization and pharmacology and physiology. And Bruce's lab is still pushing out those brilliant foundational publications. And this webinar is no different in the sense that he'll be building on a lot of that work that's come out of his lab in more recent months. Before Bruce comes on, my colleague, Mads Korsgaard, who is Global Product Manager at Sophion Bioscience. He is also a veteran of the scene. So he had 12 years of neuroresearch, where essentially as part of neuroresearch, they were some of the founders of Sophion. So he's been heavily involved in adopting and developing Sophion products since his time at neuroresearch back in the early 2000s. And since 2013, I believe, Mads has been part of the team at Sophion. Mads will be our first speaker, giving you a brief introduction, and then we'll bring Bruce on to give the main event, the star of the show, his webinar on sodium and potassium channels as novel targets for pain and epilepsy. Some really interesting work that Bruce's lab has been doing. So without further ado, I think I'll invite Mads to the stage, and Mads can take over from here and give you the introduction.
Well, thank you very much, Damien, for the introduction, and also myself would like to welcome this webinar to both the audience and not the least to Professor Bruce Bean for giving these very interesting data that you're going to see very shortly. Before that, I will, however, just dwell a little bit about what Sophion is interested in and what role we have been playing in developing these tools for doing patch clamp on a big scale. So we've been here, as Damien more or less mentioned, since 2000 and developed a lot of things over time. Pursuing the need for speed, I almost said need for throughput, while preserving the fidelity of the manual patch clamp that we know from the early days of in vitro electrophysiology. And we believe we have achieved many of those goals, but we don't stand still. We keep pursuing getting better and better. And why is that? That is, of course, because ion channels as a whole play on a rough scale a third of all known diseases that involves ion channels, and more are these years coming to surface where ion channels play a pivotal role. And they are addressed for three sort of main reasons. Identify compounds that can then modulate the ion channel's function, characterize the ion channel itself from the curiosity that characterizes human beings, so to speak. So all the scientific work going on, where Bruce Bean has been more in the forefront for many years. And then there is, of course, also the leg of adverse effects of new drugs, so ensuring that you do not have serious adverse effects, primarily known from the cardiovascular systems, like blocking a hERG channel, which would be very bad. So you can see here a little model or a little cartoon of how many disease types are around ion channels, and we will today hear much more about pain and epilepsy, and how to find new modulators, gating modifiers of these diseases. Traditionally, drug discovery has taken its offset in fluorescence-based technologies. So if you look at this funnel, it has been a matter of a high throughput screen, really trial and error through large libraries of molecules, and then having used such indirect measures as the fluorescence-based technologies mostly are when it comes to ion channels, it has been necessary to do a secondary screen or a confirmatory hit screen, as somebody also called it. We have the ambition to actually play a role in drug discovery by taking part of already at the primary screen level. And to do that, we have, of course, developed higher throughput means of testing ion channels and modulators hereof. And if you look at it in another way, what is sort of the justification of this, well, admitted slightly or maybe not so slightly a more expensive technology? The justification comes from the fact that doing electrophysiology up front gives you a much better starting material for the medicinal chemist when you deliver back the data from the molecules that have been tested in the primary screen. You, as given here in this snake-like cartoon, you don't need to do the assay development after you've done your target discovery, because part of target discovery is 𝗂𝗵𝐻. And you don't need to do the hit validation, as I already said. And then, not the least, the lead optimization is much better off with the data coming out of electrophysiology. So without dwelling more at this, I would let data speak. So I'm sure Professor Bean will address some of the functionality benefits of using real electrophysiology, the voltage clamp, the fact that you can manipulate the channel into the state that you want it in when you're looking for molecule effects. So please hand over the stage to Bruce Bean for his talk. Thank you.
Thanks, Mads. That's lovely, lovely introduction. And now to the main event, the star of the show, Professor Bean. Before Professor Bean comes on, I would like to just highlight the fact that we will have questions to Bruce and potentially to Mads too, if there are technical questions for Mads. But we'll try and address as many of these questions as we possibly can at the end. So put your questions into the chat box. We will do all we can to try and get those answered as best we can. As my colleague Jette has also mentioned in the chat, we're also having some polls that will be popping up. Please take a few moments. They're pretty simple, straightforward polls to just give us some of your feedback on those polls there. And again, thanks to Professor Bruce Bean for what should be a fantastic webinar. And I will hand over to Bruce right now. Over to you, Bruce.
Great. Thanks, Damien. Thanks, Mads. So yeah, I'm going to review some work we've been doing in my lab. My lab has gradually transitioned from doing really only basic science research in ion channel pharmacology to trying to do early stages of drug discovery. And as we have done that, we have really transitioned from starting with fluorescence-based screens to relying on initial screens using automated patch clamp. And so I'm going to review some of our experience in doing that, some of our results, and very much welcome a discussion from everybody at the end of the presentation. So the essence of what I'm going to talk about is the fact that I think most of us are very aware of if we've worked in ion channel pharmacology, and that is that the vast majority of ion channel targeted compounds don't work in a simple way by just sort of physically blocking the channel the way that tetrodotoxin does. Almost all small molecule ion channel targeted drugs are state dependent. Almost all of them work in an allosteric mechanism by either stabilizing or destabilizing particular states of the channel. And this is a concept that was introduced by 𝗱𝗏𝗂𝗏 in 1977 in his really seminal investigations of local anesthetics, and in showing that the way local anesthetics work is to allosterically stabilize inactivated states of the channel. And this is really the basis of understanding the state dependence of a huge range of other ion channel targeted compounds. And because of the fact that compounds work in a state dependent manner, it's really critical to try to capture that state dependence we have found from the very beginning, which is ideally done using patch clamp, which of course in the old days with manual patch clamp was exceedingly tedious, but now has gotten faster and faster. And I'll just say that, you know, in our experience, we started off as sort of a gateway to automated patch clamp by being able to be beta testers for the Qube compact. I was actually initially a little bit reluctant to even, you know, bother being a beta tester for that, because we have a lot of expertise in manual patch clamp. You know, we can do it fairly quickly, but we very quickly got to appreciate the Qube compact with 8-channel, being able to do 8-channel pharmacology, and that led us to an interest in moving up to the 384-channel automated patch clamping. So to go back to the very beginning of all of ion channel pharmacology, of course, Hodgkin and Huxley found that the action potential is generated by voltage-dependent conductances. The key conductance in the action potential is the operation of voltage-dependent sodium channels that are opened by depolarization and then go through a series of state changes where they inactivate with maintained depolarization. And so the fact that sodium channels can exist in these different states is fundamental to the way they work in generating the action potential. In the squid axon, for those of us who were, you know, starting our careers in the 1970s, it seemed like everything was very simple. 2 voltage-dependent channels, a sodium channel generating the upstroke of the action potential, a potassium channel generating the downstroke. But as work by many people showed, particularly in doing recordings from central neurons, we, as a field, very rapidly appreciated that although action potentials are very similar in mammalian neurons to the squid axon in their basic shape, they actually differ a great deal in the shape of the action potential and especially in the patterns of firing that you see. And really concurrently with that was the revolution in the 1980s from molecular cloning and from patch clamp that allowed us to do recordings from mammalian neurons and do voltage clamp from mammalian neurons that was previously very difficult to do. And the combination of molecular cloning and patch clamping from mammalian neurons showed us that there is an amazing diversity of ion channels that underlie the action potentials and neuronal firing in mammalian neurons. You know, far richer than you ever would have thought from the analysis of the squid axon action potential. And this has really transformed, I think, you know, the possibilities of pharmacology, because since there is such a wide diversity of ion channels in central neurons, and because the repertoire of ion channels differs from neuron to neuron, we have the capability, or the possibility at least, of going in and selectively modulating the activity of particular kinds of neurons in ways to either treat disease or or other reasons for modulating. And this is a slide I use for teaching, which is by no means complete. I sort of stopped updating it once it wouldn't really fit very well vertically on the screen. But these are channels that are really present in almost any mammalian neuron that you would study by voltage dependent, by patch clamping it. And initially, we recognized many of these channels by their pharmacology, along with molecular cloning. And I've listed here, some of the compounds that were key in differentiating these channels one from another. And I'd like to point out in green that the red compounds are inhibitors, which are, of course, the most commonly studied, the earliest studied kinds of ion channel modulators. But there are also a number of enhancers of ion channels that have been discovered. And particularly for potassium channels, as I'm going to discuss, we think that enhancing potassium channels, specific potassium channels, is a kind of pharmacology that is ripe for exploitation now that we have more information about the molecular pharmacology of particular kinds of K channels and a better recognition of how expression of K channels differ in different kinds of neurons. And as Damien mentioned, Mad's previous association with NeuroSearch was a company that was in the forefront of developing enhancers for potassium channels. And you can see here these NS compounds, particularly for calcium activated K channels. Many of them came from work at NeuroSearch. So let's see, I'm going to go forward here. Okay, so to talk about sodium channels a bit, of course, as I mentioned, these gate between different channels as a function of time and voltage. That's key to the way they work in the action potential. 𝗱𝗏𝗃𝗃 found that local anesthetics like lidocaine work by binding tightly to the inactivated state. And even though the inactivated state is non-conducting, binding of a compound to the inactivated state stabilizes that state and then removes channels from the resting state where they're available for activation. And so that's the way local anesthetics work. It turns out that you can very easily quantify the binding to the inactivated state by looking at shifts of the inactivation curve of sodium channels. Many other compounds work in this way. The compounds that work in this way produce the characteristic use-dependent property, where because the bound inactivated state returns to the resting state slowly, you can get this phenomenon of use dependence. Many of the sodium channel targeted anti-epileptic drugs like phenytoin work in this way, where they have very weak binding to the resting state, strong binding to the inactivated state. Recently, one of the compounds we got interested in was cannabidiol, which is FDA approved for treating epilepsy, 𝗱𝗏𝗃𝗃, marketed by Jazz Pharmaceuticals. And although when it was discovered, the mechanism of cannabidiol as an anti-epileptic drug was not well understood, we have now know that it's a quite potent inhibitor of voltage-dependent sodium channels and works in this allosteric mechanism, very much like local anesthetics and other anti-epileptic drugs, where it binds much more tightly to inactivated state channels than the resting state channels. So I'm going to come back to some work we've been doing with cannabidiol-inspired compounds a little bit later in the talk. Another kind of state dependence that's very unusual. So that state dependence where anti-epileptic drugs, local anesthetics, and many other drugs bind tightly to the inactivated state, that's very common. Much less common is a kind of state dependence that we found in looking at some NaV1.8 inhibitors, starting with A887-826, an Abbott 𝗃1.7 inhibitor developed by Doug Kraft and his colleagues, and more recently the VX548 suzetrigine, the 𝗃1.8 inhibitor made by Paul Negulescu and his colleagues at Vertex, which has recently been FDA approved for treating pain and seems to be very effective in a variety of pain mechanisms. And interestingly, with both A887826, VX548, Latigo33, and some others, other 𝗃1.8 targeted inhibitors, these actually have the opposite state dependence, where they bind tightly to the resting state of the channel and actually bind very weakly when the channel is depolarized. And so you can actually put on a depolarization and drive them off the channel quite effectively. If you put on the depolarization for long enough, then you can drive them almost completely off the channel. In the case of A887826, this kind of state dependence produces reverse use dependence, where even with action potential waveforms, you can drive the compound off the channel with repeated action potential waveforms and therefore decrease its inhibition. With VX548, we found that although the phenomenon of state dependence is quite similar, it requires such large depolarizations and requires long depolarizations so that there's effectively no removal of inhibition physiologically with physiological waveforms. So in the case of VX548, it should not affect the ability of the compound to inhibit the channels. But still, it reveals something kind of fundamental about the way the compounds interact with the channel. Potassium channel enhancers, which I'll talk a fair amount about, virtually all of them that I know of also work by shifting either the voltage dependence of activation or, in the case of calcium-activated K channels, the calcium dependence of activation of the channels. And a good example here is 𝗻𝗃𝗄𝗏, which is an anti-epileptic drug that acts by enhancing Kv7 channels. It has now been taken off the market because of non-targeted related issues, but a number of companies, including Xenon, are developing improved Kv7 activators that do not have these problems. 𝗻𝗃𝗄𝗏 acts by shifting the voltage dependence of Kv7 channels towards more negative voltages. And so if you do voltage clamp experiments and you look, say, at a depolarization with maximal activation of the K channels to +20, there's essentially no effect of 𝗻𝗃𝗄𝗏. But if you look at -60 millivolts, -50 millivolts, around where action potentials start to be generated, then there's a very profound enhancement of the current. And so the voltage dependence of channel activation is shifted in the hyperpolarizing direction. We have found that this same mechanism of action for Kv7 channels happens with cannabidiol. So cannabidiol, in addition to inhibiting sodium channels, we found enhances Kv7 channels in a manner very similar to 𝗻𝗃𝗄𝗏 by shifting the voltage dependence in the hyperpolarizing direction. Interestingly, it's quite a lot more potent than 𝗻𝗃𝗄𝗏 is in doing this. So now I'm going to talk about some work that we've been doing, and we here is a very close collaboration with my colleague Clifford Wolff, with whom we've been working on pharmacology for pain for many years. And this is based on trying to find novel sodium channel targeted drugs and also potassium channel targeted drugs for treating pain. And as many people in the audience will know, sodium channels have been a very prominent target for novel pain medications for many years now. There was huge excitement in 2006 with the discovery of families where a null mutation in NaV1.7 produced congenital insensitivity to pain. That led to the development of programs at many companies for developing selective NaV1.7 inhibitors. The chemistry on many of these programs was incredibly impressive in developing very quickly highly selective, highly potent NaV1.7. inhibitors. Clinically, this has so far been disappointing. There's two reasons for that. One is that many of these compounds are sulfonamides, and as it turns out, many of the sulfonamides that were developed have extremely high plasma protein binding and probably binding to other proteins in various tissues that affects their bioavailability. Another problem that was highlighted by work at Genentech and by the Merck group is that the NaV1.7 inhibition, if you have an extremely potent inhibitor, produces hypotension if you give that inhibitor very quickly. Probably doesn't happen if you ramp up the concentration of NaV1.7 inhibition, consistent with the fact that, interestingly, the human patients with the null mutations don't have problems with their autonomic nervous system. However, this problem led to the discontinuation of NaV1.7 inhibitor programs in a number of companies. Much more effective clinically so far has been NaV1.7 inhibition, and VX548 is quite a remarkable compound for its selectivity, for its potency in inhibiting NaV1.7. Quite an amazing accomplishment by the group of scientists at Vertex. Effective clinically for treating acute pain. Although, if you look at the effects on acute pain, much better than placebo. And from what I've heard from neurologist colleagues, effective in patients in a variety of pain indications. However, very far from producing complete analgesia, and I'll come back to this in a second. And so because of this, I noticed an interesting article in the New Yorker recently focusing on suzetrigine as, an incredible accomplishment in pain treatment. The suzetrigine shows that you can inhibit nociceptor excitability and affect clinically human pain. But in the interview with Paul Negulescu, he made the point that the work at Vertex is continuing to look at NaV1.7 and specifically looking at combining block of NaV1.8 and NaV1.7. On the left here, I show some data from our lab where we looked at VX548 on human DRG neurons, which were provided to us by the team at Antibios. And what we see is that VX548 at high concentrations relative to the IC50 is reasonably effective at reducing repetitive firing, but it doesn't completely block it. And because there are NaV1.7 channels that are very prominent in the neurons, they can still fire a single action potential very effectively, and in some cases continue to fire repetitively. And so because of the fact that there is prominent expression of NaV1.7 along with NaV1.8, I think it makes sense that inhibiting NaV1.8 alone may have an intrinsic ceiling, at least in inhibiting the excitability of human nociceptors. And so we think that actually goes along pretty well with the clinical data. So that got us interested, quite a few years ago, in the idea of developing combined inhibitors for NaV1.7 and NaV1.8. And that's something that we've been working with Clifford Wolf and with some collaborating medicinal chemists who I'll talk about in a second for a number of years now. So this began by doing high throughput screening using a fluorescence assay. This was developed in Clifford's lab at Boston Children's Hospital. This was based on a fluorescence assay originally described in this paper from David Weaver's lab at Vanderbilt and improved by Dan Beecham at Molecular Probes, with whom Clifford's lab worked very closely in developing this as a high throughput screen for both NaV1.7 and NaV1.8 inhibitors. It's based on the entry of thallium through sodium activated through sodium channels, and then with high output fluorescence probes for thallium. Now, one of the features here of this is that you have to prolong the activation of the sodium channels long enough to produce effective thallium flux so that the channels don't inactivate quickly. And this is done by using veratridine, which is very commonly used in these kinds of experiments, which both shifts the activation of sodium channels and slows the inactivation so that you can get effective entry of thallium. It also, by the way, veratridine changes the pore structure of the channel to make it more permeant to thallium than it otherwise would be. And so this high throughput screen with the help of Dan Beecham at Molecular Probes and with a lot of work by Lee Barrett and Jae-Hoon Chim in Clifford's lab became very effective as a high throughput screen for NAV 1.7 and NAV 1.8. Here are some examples of hits from this screen where you can see with these asterisks these compounds were effective at inhibiting the entry of thallium through the modified sodium channels. So this was the beginning of our program. As this went forward, we did, as Mads said, basically a secondary screen once we had access to automated patch clamp technology, and then we used this to try to verify these hits that we got from the high throughput fluorescence assay. And as I'll show you, some of the hits were validated and some weren't. And so this led us to do a more systematic comparison of the fluorescence assay with the results with the voltage clamp on the Qube. So here's an example. Actually, this is a fairly early example of running our NAV 1.7 cell line on the Qube. We typically now get recordings from all 384 channels quite often. Here you can see there's a few channels that didn't give good recordings, but you can see that there's very nice voltage control of the channels here of the NAV 1.7 current. This happens to be a chip using a single cell recording. We've since, for most of the pharmacology, switched to using the multi-hole chips, which works better for screening. So here are the methods for the assay using the Qube 384. This uses a NAV 1.7 cell line. As I mentioned, we've switched to using multi-hole 10-hole electrode plates for doing most of the initial screening. And the key thing here is to use a protocol where we're quantifying the voltage-dependent action of the channels. So we can use, of course, unmodified channels without veratridine, using voltage steps to activate the channels. And we can mimic the voltages that would be seen by a sodium channel in a nociceptor. And so, for example, we do that typically by changing the resting potential to either -70 millivolts, -60 millivolts, typical resting potentials for nociceptors, and then assay the activation of the channels. We do that by comparison with holding the channels at -100 millivolts, because that gives us information on the state dependence of the channels, which is very useful to get right in the first screen. And you can see here a typical example of some test compounds here. You can see in the gray, this test compound was very effective in inhibiting the channel. And then we always follow by an application with NAV 1.7 of TTX or with NAV 1.8 of 𝗵𝗁548 to verify the access and so forth to the solution change, which is almost always perfect. So here's an example of some of the recordings that we've done with a variety of existing compounds on the Qube. Here's an interesting compound, riluzole, used clinically for treating ALS. It is both an SK channel enhancer and a fairly potent sodium channel inhibitor. And as you can see, like local anesthetics, like anti-epileptic drugs, it's an extremely state-dependent molecule where there's very little inhibition of the channel from -100, but you can see at a more typical resting potential that there's quite effective inhibition of the channels. So here's a comparison. So we got interested now in, and basically what we did was to run a group of compounds from a variety of libraries, including many known compounds, on both the thallium flux fluorescence assay and on the Qube, and then compared the results from the two of them. And as you can see here, so here's the inhibition from 0 to 100 of the thallium flux on the y-axis, inhibition from 0 to 100 with voltage clamp on the Qube. And you can see that overall the correlation is quite poor. And so we dove into this a little bit more by looking at specific classes of compounds to look at this. And in particular, what you can see is that if you look at the fluorescence assay, there were quite a few basically false positives that were not confirmed by voltage clamp. And probably even more important for those of us interested in screening, there were quite a few false negatives from fluorescence. So to dive into some of the classes of compounds, in general, local anesthetics gave similar results with the fluorescence assay and voltage clamp. So they tended to be more potent in looking at the voltage clamp. And here we made use, as I mentioned, of looking at voltage clamp from using steps from -70 millivolts to be more physiologically relevant. And that's probably part of the reason that we see more effective inhibition, more potent inhibition with the voltage clamp. With the thallium flux measurement, you don't have control of the voltage in the cell. You don't know exactly what it is, and that's part of the difficulty in interpreting those results. One of the classes of sodium channel inhibiting compounds that I think is particularly interesting are tricyclic based compounds. For example, amitriptyline was originally introduced as a tricyclic antidepressant, but as work by Birgit Priest and Ivy Dick and their colleagues at Merck, I believe at the time, Birgit is now at Lilly, showed amitriptyline is also quite a potent sodium channel inhibitor. Amitriptyline is now a first-line treatment for many kinds of neuropathic pain, and it's very likely that it works as a sodium channel inhibitor. It has a structure that's actually fairly similar to carbamazepine, but is a lot more potent than carbamazepine as a sodium channel inhibitor. Another compound in this class is doxepin. And what you can see here is that in general, the fluorescence assay did pick up inhibition by this class of compounds, although for some of them, like doxepin, the results from the thallium flux were basically a false negative. Anticonvulsants in general were fairly weak in the fluorescence assay compared with the Qube assay. The biggest issue with the fluorescence assay was that it essentially fails at all to pick up inhibition by the sulfonamide class of NaV1.7 and NaV1.8 inhibitors. And so here are some of these compounds, sulfonamide compounds, the 𝗂𝘁𝗃𝗃 compound, two of the Pfizer compounds, and you can see that there's really very weak effects in the thallium flux assay, but very potent and with nice dose dependence in the Qube assay. Another compound like this is vixotrigine, the convergence compound that was brought into clinical trials at Biogen, and again, especially at lower concentrations, this was very weak in inhibiting the thallium flux in the fluorescence assay, but very effective in the Qube. Now moving on to NaV1.8, which we also studied with these libraries. These were done on two different NaV1.8 cell lines, a CHO cell line, a HEX cell line, again, multi-hole chips. Here's an example in gray of a hit from one of the libraries. And again, we're using voltage clamp protocols to try to assay physiological action of the compounds. Here, we're particularly interested in inhibition at more depolarized voltages, because the role of NaV1.7 channels is largely to enable repetitive firing by not inactivating at -40 or -50 millivolts where NaV1.7 channels activate. And so here we looked at channels at voltages where the availability of NaV1.8 channels physiologically is important at -50, -40 millivolts. Okay, so some of the this is actually comparing two different cell lines, a hex cell line and a CHO cell line. The CHO cell line is the one we prefer. It has a beta-3 subunit co-expressed with the NaV1.8. And you can see there's excellent correlation really with the effects of all of these compounds, all these reference compounds for the two cell lines, both run on the Qube. Now, when we compared these 2000 test compounds on the fluorescence assay and the Qube voltage clamp, there was extremely poor correlation in the results. And one of the most striking things actually was that many of the compounds actually enhanced the thallium flux fluorescence signal. We don't really understand why, but quite a few compounds did that, showing up basically as negative values for inhibition here. And that is something that was never confirmed using the Qube. Again, some of the specific compounds that we looked at with thallium flux on the Qube, where there was very poor detection by the thallium flux, included, for example, this 𝗲𝗁 compound. So to summarize here, basically at this stage in our drug development program with Clifford's lab, we switched from doing things even in the beginning with thallium flux to doing the screens from the beginning on the Qube. And the reason is because there was such poor correlation, and especially because of the false negatives coming from the thallium flux, which is very concerning because you don't want to miss promising compounds. And as I mentioned, particularly with the sulfonamide compounds, there was very poor detection of both NaV1.7 and NaV1.8 inhibitors with the thallium flux. So with that background, I'm going to show some of the data that we've gotten in trying to develop dual inhibitors of NaV1.7, NaV1.8. I think I've already discussed really the advantages of the Qube 384 here. Most prominently, that the initial assay, we can run any voltage protocols we like. We can build in a test for state dependence. We can quantify the state dependence even by using a variety of holding membrane potentials. And you can see for amitriptyline, again, very strong state dependence. Riluzole, very strong state dependence. In the case of the sulfonamides, there actually is state dependence, but it's at such a time scale that once the channels are inhibited, they essentially show up in the assay, depending on how you structure it as showing weak state dependence. We can talk more about that later if people want to. We typically build in actually use-dependent protocols that we run even in the initial assays. And you can see here, these are very useful because you can see that for amitriptyline, you get fairly strong use-dependent inhibition of the channels, but with A887826, we can see this relief of inhibition in a use-dependent manner. So now I'm going to turn to talking about some of the results. I'm going to sort of skip over a little bit, some of the early stage medicinal chemistry and so forth. This was a series of over about 3 or 4 years of medicinal chemistry done by 𝗂𝗰𝗄 and Mark Namchuk, very experienced medicinal chemists. Again, close collaboration with Clifford Wolf's lab. And I'll just show some of the highlights. What we ended up doing was exactly what Mads referred to is we ended up doing a cycle of medicinal chemistry followed by characterization on the Qube, testing dose response, state dependence on the Qube, and then going back and doing optimizing medicinal chemistry. And what we were trying to do here is to find compounds optimized that were both NaV1.7 inhibitors and NaV1.8 inhibitors. We decided in doing the medicinal chemistry to try to optimize the NaV1.8 inhibition based on, even before the FDA approval, on knowing that there were promising results with the Vertex program with NaV1.8, possible problems with very potent inhibition of NaV1.7. And so our strategy was try to optimize NaV1.8 inhibition, have some NaV1.7 inhibition, but not as potent. Here's an example of one of the compounds that Corey and Mark designed based on many iterations over a number of years of testing on the Qube and doing medicinal chemistry. This compound, you can see, works really nicely, showing dose dependence inhibition of NaV1.7, stronger potency on NaV1.8. You can see here, the DMSO controls work really well in these Qube assays with very little inhibition. Excellent results with the positive controls, either TTX, 𝗱𝗵𝗃𝗴, and we've really worked out the protocols where we're getting a really very high quality data in a very reproducible way, and that's greatly enabled this program. Here is another compound where, again, we're trying to optimize NaV1.8 inhibition, have some NaV1.7 inhibition, and we have then, in collaboration with Clifford's lab, gone on to test these in mouse pain models, which I'll show you in a second. So the second part of our screening strategy here is, once we have tested them on the Qube for NaV1.8, NaV1.7 inhibitors, we look at their effect on reducing excitability of nociceptors using acutely dissociated mouse nociceptors. These are a mouse line with CGRP promoter driving the expression of fluorescent indicators, so we're identifying nociceptors. And you can see that several of these combined NaV1.8, NaV1.7 inhibitors are very effective in inhibiting repetitive firing in the mouse nociceptor assay. Next, we've progressed some of these into looking at the effects on pain behavior in Clifford's lab. We typically start with a hot plate test, very easy to do. Here's an example of one of these compounds that is, not the most potent, but quite effective in inhibiting pain behavior on the hot plate test. We next move to a really nice system that has been developed in Clifford's lab called the black box system, in which an animal is freely moving in the dark and is assayed for its behavior, for the pressure it puts on a paw in real time during natural behavior. So far more physiological than using von Frey hairs or other stimulus evoked behavior. And this has really been transformative in being able to look at a large variety of compounds quickly. We can see here this compound was quite effective at 10 mg/kg in reducing, and even at 3 mg/kg actually, in reducing nociceptive behavior, and up to 10 mg/kg did not produce sedation. Unlike, for example, amitriptyline, that actually produces sedation even at analgesic concentrations in this system. The other program that we've had, which is actually similar in that we're looking for combined NaV1.7, NaV1.8 inhibitors, is a program that originated from our discovery that cannabidiol activates Kv7, as well as inhibiting NAV channels. And we found that cannabidiol inhibits NAV channels fairly non-selectively. So it inhibits NaV1.7, NaV1.8, NaV1.6, and so forth, including NaV1.5, actually, which is something that we could talk about, fairly effectively, and also enhances Kv7 channels. And so we thought that the combination of inhibiting both NaV1.8 and NaV1.7 and also enhancing Kv7 would be very promising for developing novel pain-targeted compounds. And so this is a work done in collaboration with a very experienced medicinal chemist, Jed Hubbs, at Boston Children's Hospital over the last 4 or 5 years. This program, again, started before this timeline with some screening with thallium flux, but as soon as we could, we moved to doing everything on the Qube, and that's what's shown here. And so basically what we've been doing is very quick cycles of medicinal chemistry and then testing for inhibition of NaV1.7 and NaV1.8 on the Qube, activation of Kv7 on the Qube, and looking for compounds that are more effective than cannabidiol for these effects. So the effect of cannabidiol on the Qube is shown here by these red lines. And so what we're looking for are things that are more effective at inhibiting NaV1.7 and NaV1.8 and more effective at enhancing Kv7 than cannabidiol. You can see that in the initial series of compounds that we studied, we had some that were more effective in inhibiting NaV1.8, a few that were a little more effective in inhibiting NaV1.7, but we were not able in this series of compounds to retain the Kv7 activation. And so as we have gone along, and again, greatly facilitated by being able to do these cycles of medicinal chemistry, test them on the Qube, very quickly, and then go back and do another cycle. And as we have gone along, we've been able to enhance the Kv7 activation, enhance the NaV1.8 inhibition, and then with the compounds shown in red, we've progressed these to studying their effects on nociceptor excitability, and then in these compounds, in a subset of these compounds, gone on to test them in Clifford's lab on pain behaviors. This is very much an ongoing project, although we are very excited about this because we found a number of compounds. For example, this compound here, it's quite an effective Kv7 activator. It's a very effective NaV1.8 inhibitor. And if we look at this, again, triple acting compound, NaV1.8, NaV1.7 inhibitor, Kv7 activator, it's quite effective behaviorally in inhibiting, for example, nociceptive behavior Paul linking. We've also looked at this on human DRG neurons and find that it's very effective at inhibiting the excitability of the neurons. I want to close by talking about really what we're most excited about in the lab now, and that is developing novel K-channel enhancers for both pain and epilepsy. And I'm going to, the time is too short to go into all of this, but I'll give some of the highlights here. So one of the targets we chose was Kv1.6. And this is based on work that we did with David Ginty's lab, looking at both electrophysiology and mRNA expression in different kinds of DRG neurons, and finding very prominent expression of Kv1 channels, and specifically Kv1.6 channels in nociceptors, in most nociceptors. Kv1.6 channels is also strongly expressed in DRG of human neurons. The interesting thing about Kv1 as a drug target is that Kv1 channels are activated by small depolarizations, subthreshold depolarizations. And so they're very effective at producing a brake on excitability. So, for example, you can see this very nicely in neurons where Kv1 is expressed highly. Often these neurons will only fire 1 or 2 action potentials. because of the activation of Kv1. If you inhibit them, for example, with alpha dendrotoxin, then you can convert firing of just 1 or 2 action potentials into very strong repetitive firing with a lower threshold. So the idea is, if you have this kind of repetitive activity in nociceptors to begin with, or you have hyperexcitability as a result of changes with neuropathic events, then by enhancing the Kv1.6 activation, we think we can convert the repetitive firing to firing of just 1 or 2 action potentials. And so here's our drug discovery strategy for this program. We are screening, directly onto the Qube 384. We've started off. We haven't yet screened all 160,000 compounds, as I'll discuss at the end. We have been rudely interrupted in this program by the withdrawal of our NIH funding. But before that happened, we got through 25,000 compounds and already have some hits that we're pursuing. So we test them directly with primary screen on human NaV1.6 on the Qube, then do dose response on the hits, then go to studying them on mouse nociceptors, and then onto human DRG neurons, and then do counter screens on undesired targets. So I'm just going to go through some of the highlights here. So here's an example of recordings from a screen with Kv1.6. We started off here with a positive control compound that we took from a paper by Frederick Ellender's group, where they did small-scale screens, relatively small-scale screens on shaker K channels, and found a number of compounds that enhance the opening of cloned shaker K channels by increasing coupling of the voltage sensor to the opening of the channel. Not all of these worked on Kv1.6 channels, but some did, and one that was reported in their paper that we found to be a very useful tool compound we call KVN, KV enhancer 06 at 10 micromolar, you can see, is fairly effective at producing enhancement of the current. And as you can see, this changes the kinetics of the channel. It basically slows the deactivation of the channel at -40 millivolts. It's a very state-dependent effect. And so one of the features of these is that some of them enhance the current so much that you can actually get enhanced current -80 millivolts around the resting potential of the cell. And so here's actually somewhat dated, but an early version of the primary screen that we did on 25,000 compounds, where we found a number of, this is the positive control, we found a number of enhancers. There's many more inhibitors than there are enhancers, but we detected quite a few enhancers also. Here's an example of one of the enhancers in the primary screen, again, sort of a small scale screen, 25,000 compounds, quite effective. In blue, the positive control, the KVN006 is here in red. That's shown in this column. You can see the very good reproducibility in the wells in this column for the positive control, negative control here in this column, and it's made it very obvious, the hits here. These hits, all of the hits we have so far, work by shifting the voltage dependence of the channels. And so this is something that you would not have seen, for example, by stepping the voltage to 0 millivolts here. There's really very little effect of the compound. It works by enhancing the opening of the channel at negative voltages at -40 millivolts, and here, very strikingly, a -80 millivolts near the resting potential of a cell. This particular compound has very good selectivity over other K channels that we're running screens on, including Kv2.2, Kv3.1, which I'll come back to for a second. We found that this compound is effective at inhibiting nociceptor excitability, both in mouse nociceptors and, at least in some cases, in human DRG neurons. We're also very interested in Kv3 enhancers. And the reason for this, and I won't go into all of the background here, but there's evidence that enhancing Kv3 channels may have utility in epilepsy, in work from 𝗁𝗂𝗂𝗂𝗂, who has been developing a number of these in Fragile X syndrome, in Alzheimer's disease, and interestingly, in the most recent patent applications from the group at 𝗁𝗂𝗂𝗂𝗂 in pain. And the reason is that Kv3 channels are actually heavily expressed in nociceptors. So we have this, for us, has been the most productive screen of all potassium channel enhancers. We're finding lots of novel Kv3.1 enhancers. Here's an example of one of these hits. And again, this has been a small scale, but already we have lots of hits, some of which are just as effective and just as potent or even more potent than the autophony tool compound 𝗁1, which we used as a positive control. And we're now very interested in moving these forward and testing them in various epilepsy models, and of course in doing medicinal chemistry to optimize these. So I hope there's going to be plenty of time for questions. I'll conclude here. The work in my lab has been carried out by 𝗂𝗃 Fujita, Tommaso Sorno, Robin Stewart, some of the early work by Patrick Valley, who got so enthusiastic about the work with the Q-patch compact and the Qube that he actually moved to Sophion, where he is now remains a key member of our team, and working out the protocols and so forth. But from the Sophion side, we have benefited enormously over the years from a very close collaboration from a screening core at Harvard Medical School led by Jen Smith. This has been fantastic for using their technology for generating the plates for the 384 channel screens. As I mentioned, a very long collaboration on pain with Clifford Wolf's lab, and particularly with Jae-hoon and Lee Barrett in the NaV1.7, NaV1.8 drug discovery. The medicinal chemistry and the NaV1.7, NaV1.8 drug discovery was by 𝗂𝗰𝗄, and I should have listed Mark Namchuk here, also Jinbo Lee and Jed Hubbs on the CBD derived compounds. We've had a great collaboration with 𝗂𝗵𝘃𝗆 in being able to test some of these compounds on human DRG neurons, and wonderful back and forth in developing the protocols and optimizing things with the group of scientists at Sophion. The work was, until last March, funded by an NIH grant, which was terminated with extreme prejudice against Harvard, which remains, and it's been really helpful in being able to continue our work, having support, both moral support and instrument support, and cells from 𝗂𝗵𝘃𝗆 and support from Sophion, and being able to continue the use of the Qube 384. So I'll stop here so that we have plenty of times for discussion, because I'm sure there'll be lots of things to talk about.
Thank you, Bruce. That was brilliant. I mean, it's a real whirlwind from the Hodgkin foundations and your own work in the early days in the 1980s on sodium channel activities, right through to some of your great sort of pharmacology and almost sort of drug development, drug discovery style recordings that you've been doing most recently. We do have quite a number of questions, so we'll try and answer as many as we can. Professor Bean is willing to stick around generously to try and answer as many as we can. We probably will have to cut it off at some point, because people like Professor Bean have got a lot of busy sort of work on their hands, so we can't be here all night, but we will do what we can. Bruce, what might help is in the chat box, if you go and have chat highlighted on the far right of it, you can see a question mark with a number 13 beside it. That, if you click on that, that should give you all the questions. I will read them out, but it's often easier as well if you might be able to sort of follow along. The first one from Ashutosh has been answered. The next one down from Ludmila is what 𝗂𝗶7.2 cell line was used?
𝗂𝗶7.2 cell line.
Hmm.
That is a great question. We have used multiple, two different 𝗂𝗶7.2 cell lines, a CHO cell line and a HEX cell line. They both work. Both have been a bit challenging, actually, compared with all the other K channels. And we're actually in the process of optimizing those cell lines with some new protocols that we're doing together with Patrick Valley at Sophion. So we can, I'll be glad to, if you email me. I don't have the source. I think the first cell line we got was from BSIS, if I remember correctly. The second cell line we got was from Chantest, maybe. I forget exactly the hex cell line. So anyway, if you email me, I can give you the source of those.
And working down the question.
Those are 𝗂𝗶7.2, 7.3 cell lines.
Yeah. Just working down the questions, Bruce. The next one down is Esteban. Great presentation. Have you counter screening? Have you done any counter screening for your compounds versus other NAV channels? It would be interesting if your compounds are selected against NAV 1.1, 1.5, 1.2, or 1.6.
Yes. Yeah, it's a great question. We have done some. We stopped doing that routinely, simply from limitations of sort of time and effort. And a point I wanted to make, and I didn't really have time to build this into the presentation, is this. I think that what we discovered in doing this project was that I think it is a mistake to worry too much about optimizing against NAV 1.5. And the reason for that is that we have found that virtually all clinically used, clinically tolerated sodium channel inhibitors inhibit NAV 1.5 as well or better than they do the other sodium channels. And a great example of this is amitriptyline, which is very effective at inhibiting NAV 1.5. It's used clinically for neuropathic pain. It's tolerated. There are not cardiac side effects, at least without an overdose. Another example is cannabidiol, taken by kids for treating their epilepsy. And it inhibits NAV 1.5 more than it inhibits most of the other sodium channels. Okay, so why are they clinically tolerated? The reason is the voltage dependence. The reason is that cardiac muscle sits around minus 88 millivolts, very near to EK. And because of that, any strongly state-dependent inhibitor is okay, because it won't inhibit the sodium channels, the NAV 1.5 channels, physiologically from the negative resting potential in cardiac muscle. And so if you simply throw out, the compounds that have any NAV 1.5 inhibition, I think that's an error, because I think the reason that many of these compounds are effective clinically is the strong state dependence. So a nociceptor, will sit at minus 60 millivolts, even minus 55 millivolts. You get very strong state-dependent inhibition there, and you can have intrinsically inhibition of NAV 1.5 without being a clinical problem. So, to answer the question, with some of these, we did counter screen against NAV 1.1, 1.6 early on. We have sort of de-emphasized that as we've gone forward, but we're going to come back to it because for the pain-targeted drugs, probably you want to minimize NAV 1.6 inhibition, because probably that's going to give you a little bit of sedation. But that's a little bit of a secondary issue. And for us, the strategy has been, because we have the ability with our collaboration with Clifford Wolf's lab to quickly test these in vivo, that's where we like to go. because what we want to know is it going to produce sedation? And we can look at that very directly by looking at the pain. So with some of these, we've counter-screened. We started off counter-screening against NaV 1.5, 1.1, 1.6, but we've kind of de-emphasized that most recently.
Thank you. Next question down, Bruce. Suleiman asks, is there a big difference in potency of the dual inhibitors on human versus mouse 1.7 and 1.8 channels?
That's a great question. Probably not. For most of them, probably not. We do all of our Qube runs with human channels because ultimately we want things that are going to be effective in human. And so we have not, systematically compared with mouse channels. However, our secondary test is on mouse nociceptors. And so most of the compounds that we've found are as effective on the mouse as they are on the human, which is what we do the initial screening on. If anything, many of them are a bit more effective on the mouse than the human. Now, the counterexample of that, of course, is VX548, which has very little efficacy on mouse or rat NaV 1.8 because of specific residues in the voltage sensor region that they're targeted to. But for the more conventional small molecule conventional state-dependent molecules that we've been looking at, they are as effective on the mouse and the human, which makes the preclinical studies a lot easier because we can get information from the mouse pain models. And we can also study the human DRG neurons, and in general, we see fairly parallel effects.
Thank you. And the next two questions were answered during the talk in the chat box. So I'll move on to Ed's question. Have you put any of these dual inhibitors on DRG? Based on your data showing nanoamp currents in DRG with 100x suzetrigine and action potential firing, this is really exciting.
We have not been able to do that yet systematically. So we have tested some of them on human DRG neurons. Some of them have been fairly effective at inhibiting, but we have not done that on a very wide scale. And part of the reason for that is that we, have been following up our work with the NaV 1.8 inhibition in the human DRG by looking at NaV 1.7 inhibition by selective compounds, and then looking at combinations of selective NaV 1.7 and NaV 1.8 inhibitors, rather than testing the novel ones directly on the human DRGs. But that's certainly something that we plan to do, particularly with anything that we think has potential for, further medicinal chemistry towards a true lead compound.
And I just want to check, Bruce, you're still happy to answer questions as long as
absolutely. I'll
okay.
If I give a seminar, I'm happy to stay and answer questions as long as
we'll keep going until the internet runs out. Yeah. So the next question was answered in the live chat, and then Ed asks his second question. Lovely kinetic work on VX548. VX993 recently failed in clinical trials. I have not seen the kinetic block reverse use dependence for this new molecule. Latigo VX548, meanwhile, has been published, obviously. Do you think this mechanism of action is important for clinical efficacy dampening of neuronal excitability in pain, or just a product of producing a selective NaV 1.8 inhibitor?
That's a very interesting question. Let me take a couple of points here. One is the VX993, which was the follow-up, as probably some people are aware, essentially had very disappointing results in the phase 2, which were just announced a week or so ago, and I think it has been said to be discontinued by Vertex. VX993, the structure has not been disclosed. I want to choose my wording carefully here. We obtained a compound which we believe is VX993 based on patent literature. We have not confirmed that it is VX993, so I'm not sure about that. The compound we have is strongly state dependent in exactly the same way that VX548, 𝗏𝗂33 are. So it's clearly the compound we have that we believe is VX993 has the same state dependence. We don't think that the state dependence isn't important for the clinical action. Yes and no. The tight binding to the resting state is what is important there. The fact that the compound binds to the resting state and allosterically, stabilizes the resting state and impedes activation of the compound, impedes opening of the compound. That's why it works. The knockoff of the compound by prolonged depolarizations is, if anything, a problem. And with 887, it is a problem because you can knock it off with physiological voltages. With both 𝗏𝗂33 and VX548, it's not a problem because in the case of VX548, it is not knocked off physiologically by action potential size depolarizations, action potential size voltages. With 𝗏𝗂33, it was kind of interesting because actually it is knocked off quite effectively by action potential waveforms, but it re-inhibits very quickly. And so you don't get use dependence because it re-inhibits so quickly that at physiological rates of firing of a nociceptor, it probably is knocked off, but then comes right back on again. So it's interesting because there's two different reasons, I think, that you do not get, sort of physiological use dependence with VX548 and 𝗏𝗂33. So I think it's something that has to be tested, with each one of these compounds. And we don't understand why it's so very different in kinetics and voltage dependence between 𝗏𝗂33 and VX548. They're clearly binding at the same binding site. And in mutation work that's been published by John Gilchrist and Brian Moyer at 𝗏𝗂 and by Jeremiah Osteen and his collaborators at Vertex, they're binding at the same binding site in the voltage sensor. That's clear. And I'm sure that's true for VX993, and I'm sure it's true of VX887. But the kinetics with which they come off and on are extremely different for all of them. And that's something that really needs to be looked at for each one to know if it's going to be a problem or not.
And along similar lines, actually, Marin asks, Professor Bean, you mentioned that stimulation destabilizes the drug binding to the channel. To what degree would this make it difficult to study ion channel behavior, for example, using the Markov state approach?
Oh, that's another great question. It's hard, and we have been working for a number of years on Markov modeling of sodium channel gating. And as people are probably aware, the molecular understanding of sodium channel gating, and especially of sodium channel inactivation, has been evolving recently very quickly. And initially there was the idea that inactivation was this IFM motif of an intracellular linker physically plugging the channel. Ning Yan's cryo-EM structures show that that was not true, that the IFM motif binds next to the pore but not in it and stabilizes the closed state of the pore from the inside. More recent work, there's a couple of beautiful papers from 𝗵𝗂𝗶𝗍𝗂's group with more detail about how the IFM motif stabilizes the closed state of the channel. Now, if you try to take all of that molecular data and incorporate it into a Markov model, we've been trying that. It's really hard because all of the movements of all of the 4S4 movements are different. As 𝗵𝗂𝗶𝗍𝗂's lab showed, for example, the kinetics of the movement of domain 4s4 is slowed compared with the other ones. And so when you try to incorporate all of that data into a Markov model, it gets really, really difficult. And if you try to, on top of that, add the state-dependent binding and unbinding of a compound, as far as I'm concerned, personally, we've just decided, okay, too complicated, we're not going to do it. And we actually had a student, very sophisticated computer science student working in the lab over the last half year or so, trying all the best matrix formulations of these Markov models, hoping we could get something where it was going to be accurate enough, fast enough to apply to pharmacology. But I think that's an extremely challenging problem.
So we now have two more questions in the actual marked questions that came in. I've just been looking at the chat that's been going on since, and there's a few more there. So I'll go to the marked ones, which came in earlier. So two from Ashutosh. One is there any protocol to filter out NaV1.8 currents from whole sodium currents in a DRG neuron without using TTX?
Good question. Yes, you can do it in a somewhat crude but reasonably effective way by inactivating the NaV1.7 channels with a depolarization to about minus 40 millivolts. And you have to adjust that a little bit depending on exactly what your solutions are and the details of the cell, slightly different in human than mouse. But basically, you can find a voltage around minus 40 where there's almost complete inactivation of NaV1.7 channels, but the NaV1.8 channels are only a little bit inhibited. So that's an alternative to using TTX. The problem with doing that, of course, is you cannot then look at the voltage dependence of the NaV1.8 inactivation, because if you hyperpolarize a bit, then you restore the availability of the NaV1.7 channels. So that's one way of doing it, and we do that a lot, as sort of a crude way of distinguishing components from NaV1.7 and NaV1.8.
And the second one from Ashutosh, and this will be the last of the marked questions, but as I say, depending on your time, Professor Bean, and we are going over, and I realize, your time's pretty precious.
I love this stuff. I'm happy. This is great.
Right.
I'm happy to.
Well, we've still got. I was just looking at the the people counter in terms of how we've still got 60 odd people online, so we've still got nearly 50-60% of the total that we had to start with. So there's still a lot of people tuning in to listen to your insights. So we'll keep going while we can. So the second from Ashutosh, and then we'll go to the questions that have come in since we started answering the original set of questions. So Ashutosh's second question, please discuss about why allosteric modulators are on high demand these days after suzetrigine FDA approval, and what should be the rationale for designing and screening these allosteric hits specifically for sodium channels?
Yeah, so I would say it's an experimental fact that these inhibitors, the inhibitors that are most effective, are allosteric modulators. That's just that's, I would say, something we have all discovered experimentally. Now it's possible in principle to make compounds that are not allosteric modulators. And site 1, for example, was originally, I don't know anything about their NaV1.8 inhibitor, but originally their research program was making NaV1.7 inhibitors based on Justin Dubois' technology of making TTX and STX derivatives, doing the very challenging organic chemistry there. So in principle, you could make pore blockers. It's not impossible to do that, but it's a simple fact that when we have done our screens, the compounds we have found are almost all allosteric modulators, the ones that we've sort of followed up. So, you know, you can start with the TTX and modify it, and you can make things that are pore blockers. And there are, of course, pore blockers known for calcium channels too. But as a matter of experimental observation, we find very few of those in de novo screens of small molecule drug-like libraries. And I think that's the key thing, because if you start with a sort of conventional library that people use these days for doing drug discovery, you know, you're starting with relatively small molecules that have been optimized to have drug-like molecules. Well, that's not TTX, you know, that's not STX, that's not these giant alkaloids that are pore blockers. And so I think that it's just a reflection of the fact that there seem to be very few kind of small molecules that work by really binding in the pore. Some of them might bind to binding sites that are sort of near the pore, but they still seem to be primarily allosteric mechanisms.
And we now go back to the main chat box, Bruce. So these are questions that have really come in after we started doing the marked ones. So Larry asks, great talk. Any interest in proteomic analyses, either with single cell proteomics or chemoproteomics?
That's a great question. It's really beyond, you know, anything that we are working on directly. I think it's, you know, I think it's in general, sort of for ion channel modulation, this is a really interesting area. People are probably aware of the, you know, major discovery of the way that beta adrenergic stimulation enhances cardiac calcium channels by, you know, which was basically enabled by proteomic discovery of these rad proteins that turn out to be key for regulating calcium channels. So I think it's something where there's a lot to be discovered. It's not something we are actively working on. We did a little bit of work on this together with Clifford Wolf and Peter Sorger's lab when we were initially, you know, thinking about looking for novel ways of attacking some of the ion channels involved in the pain sensing neurons. It wasn't easy to do, and we are not, at least in my lab at the moment, continuing this. But I think it's a very interesting avenue of basic research, I think.
And 𝗱𝗏𝗄𝗏, somewhat going back to something you were just talking about, Bruce, in terms of small molecules and, you know, some of the capabilities for them to get selectivity or even different mechanisms of action based on sort of sequences and what have you. So thank you, Professor Bean, really informative work and great talk. I'm curious about whether you've been able to find potent peptide enhancers or inhibitors of NAV or Kb channels in your screens. Do these peptides have unique characteristics, if any?
Another great question. We have not looked at peptide libraries at all. So we have been focused on using libraries that we, you know, selected to be kind of conventional small molecules, and you know, that was simply based on guesses that that these are likely to be things that can be developed into orally available drugs, and so we have been focused on that. People are you know, if you're interested in this and you don't know about it, there's some beautiful work done by the group at Amgen, where they specifically started with some of the known peptide inhibitors of NaV1.7 channels, and were able to develop novel peptides based on understanding of the modeling of the interaction of these with NaV1.7 to develop even more potent, you know, novel peptides based on the venom-based peptides. So I think it's something that can be done. I'm not the person to ask about, you know, there's always the question of, well, you know, how do you administer these? You know, can you ever get systemic exposure? as a way of developing an orally available drug, I think there are major challenges there. So that's the reason that we started with the conventional sort of drug-like small molecules in our screening.
And then Gary asks, the focus here is sodium block, potassium enhancement, but any effects on CAV channels involved in nociceptive pathways looked at?
Yeah.
For example, CBD might have additional useful effects on CAV 3.2.
Yeah, again, a really great question. And it's something that we have had on the books for testing for many of the compounds. And we have not yet done that because I agree, that there is in general, often some crossover, particularly between sodium channel inhibition and calcium channel inhibition. That can be a good thing. I think that if you look at many of the anticonvulsants, they're pretty non-selective again, between sodium channels and calcium channels, and that may very well be part of their efficacy clinically. So we have not yet pursued that. We on the books was particularly for CAV 2.1 channels, N-type calcium channels, which of course are a proven target for pain. With 𝗂𝗏, we have a cell line for it, and we would like to run it, against some of these compounds, and even have thought about doing a dual target with that and sodium channels. But so far we, just from resource limitations, we have not pursued that. But I think it's a great idea. And of course, as you alluded to, there's a really nice body of literature on CAV 3.2 being involved in pain. And that may be a very interesting target also.
And then Yuha asks, any concern about the KV enhancers that already work at or near to the resting membrane potential of -80 millivolts?
Concerns about them? I guess you're thinking concerns about producing excessive activation, maybe. I think it is a concern. I mean, with all of these allosteric inhibitors and all of the K channel enhancers in particular, I think we as a field, at an early stage of understanding the pharmacology, basically what happens if you activate, let's say, KV1 at -80? What happens if you activate SK channels so strongly that they are open at the resting potential? Good thing or bad thing? It's really hard to know. And it's going to depend on how things are different in inhibitory neurons versus excitatory neurons, nociceptors versus central neurons. I think it's just something that has to be addressed, trial and error pharmacology. And I would say with a lot of the K channel enhancers, really our goal at this point is, although we're going into this for a reason, that these are expressed in nociceptors, and we think in principle this could be a good target. It's very much a situation where we need to, get tool compounds that are very effective at either inhibiting or enhancing K channels, and see what they do, to the neurons and to the animals.
And Mark with a C, so I think this might be Mark Rogers. I'm really enjoying this, thank you. My question is about epilepsy. Bruce mentioned this in his talk and title, but it's not clear if he's using DRG neurons and mixed NaV1.X blockers, Kb7.X openers as a model of anti-epileptics, or just for pain. Ultimately, do the two disease indications have interchangeable features which might allow for further drug repurposing?
Another really great question, very appropriate to what we're thinking about. We have been focusing on pain, and the main, one of the reasons is that that because of our collaboration with Clifford Wolf's lab, that has been our focus. there is absolutely crossover between compounds for epilepsy and pain. You know, these are both some kinds of epilepsy, at least, you know, are basically neuronal hyperexcitability of either pain sensing neurons or central neurons. And cannabidiol is a good example where it inhibits pain sensing neurons, but also inhibits, you know, central neurons. So we have been focusing on, in our programs, on NAV 1.8 as the sort of key thing in doing the next round of medicinal chemistry. We would love to do the same thing for persistent current from NAV 1.6, which I think is probably the key target for cannabidiol as an anti-epileptic. And we've been doing a little bit in that line, too little to talk about here, but it's absolutely something that interests us, and I think it's an avenue that is going to be effective.
And reading the room very well, 𝗂𝗄 has pretty much followed up on some of what you were just talking about as well. So following on from Mark's question, maybe also other diseases linked to hyperexcitability.
Yes, right. I mean, so one of those diseases that I think is really interesting is ALS, where you know, there is, you know, reasonable evidence that an fairly early in ALS, there can be hyperexcitability of motor neurons, you know, particularly, of a variety of, not all, but some motor neurons. And in work done actually in 𝗂𝗏𝗏𝗏𝗏's lab by Brian Wenger, who now has his own lab at MGH, found that that at least with iPSC-derived motor neurons from patients with ALS, the cell death of those neurons could be delayed by 𝗂𝗶7 activation using 𝗻𝗁𝗃𝗃𝗃. And Brian and other people are now doing clinical studies with 𝗂𝗶7 enhancers for ALS. And as I mentioned, riluzole, which is, you know, been for a long time the only thing available for ALS, is a sodium channel inhibitor as well as this SK channel enhancer. So it all definitely fits with the idea that decreasing hyperexcitability, if you can get early enough in ALS, you know, could be an effective way of delaying progression of disease. And obviously, you know, there are many other diseases that might involve hyperexcitability of particular kinds of neurons. And Alzheimer's disease is another case where there's starting to be evidence of particular kinds of interneurons whose activity might be beneficial
I think that is a great place to finish, Bruce. And it's lovely to hear Brian Wenger as well, because he was working as a young MD PhD student in Steve Siegelbaum's lab when I was at Columbia. So it's great to hear that Brian's still very much involved in the field, doing great work on ALS. And incidentally, we've done some nice work on iPSC-derived patient cell lines from ALS patients. So, you know, there's some really nice work out there on that, almost as you're suggesting. That could be another avenue in terms of these hyperexcitable style diseases, or the causes or the generation of some of these diseases in the early phases of them. So thank you, Bruce. That was absolutely brilliant. I mean, a fantastic webinar, and so generous of you to stick around for, well, pretty much 40 minutes now. So we've had almost as long with the Q&A as you gave for your talk in the first instance. So very, very kind of you to stick around so long. And I think there were some really nice discussions there too. So thanks very much to you. Thanks to my colleague Mads for introducing all this at the start. And thanks to all you for staying on and listening in for so long. There's still nearly 50 odd people online listening to all your insights, Bruce. So it's been well received.
Thanks very much. Thanks for everybody for coming. It's been a pleasure interacting with everybody.
Thank you, everyone. That was great. I really appreciate all of your input, and particularly Professor Bean's webinar and taking so long to answer the Q&As. with that, we'll wrap it up. Thanks to everyone. If you do have follow-up questions, please just contact us. We will try and do our best to answer them.
Yeah.
And if we can't answer them, we'll pass them on to Bruce.
Yeah, I would say everybody should be feel free to email me. You know, I think there are a lot of people who have insights into a lot of this that go beyond what we know about, and I would love to hear from anybody who's got questions.
Thanks, everyone. Take care and goodbye.